So we had a discussion about the respiratory system introduction, and subsequently you had classes on the respiratory system. So today's topic assigned to me is about the neural control of respiration, how the nervous system controls the respiration. To deal with that, you should know about the basic organization of the central nervous system. So since central nervous system is not discussed to you so far, I just want to give you a rough idea about the central nervous system. This is an important flow diagram which you can note down and then copy. And whenever there is any question that is asked in the central nervous system, you can make a mention. So to briefly tell you about the basic organization of the central nervous system, sensory information from various receptors, for example, receives the sensory information from the skin, the fascia, joint, the skeletal muscles, and the special senses will go to the brain. Similarly, you have the sensory information from your viscera, namely the stomach, the lungs, and then the various organs in the urinary system. All these things will send sensory information to the sensory afferent division of the nervous system. And this transmits the information from the periphery to the central nervous system. Mainly all these things are by the receptor. Receptors are something like a reception of the system. So all these receptors carry the information. These details of the receptors and all that will be dealing with when we are talking about the nervous system. So the sensory information goes to the brain. That is the brain and the spinal cord. And the brain and the spinal cord will have the efferent nervous system. To remember, afferent nervous system, whenever the term afferent nervous system is discussed, that means it is the sensory nervous system which terminates into the brain and the spinal cord. Efferent nervous system is that it originates from the brain and the spinal cord and transmit the information from the central nervous system to the rest of the body. So from there it is outflow. This is an inflow, and this is not outflow coming from the motor division. That is, it transmits information from the central nervous system to the rest of the body. And this is about, for example, you have a pain stimulation to your leg, and then suddenly something pricks your sole. So that sensory information from the receptors are carried by the receptors to the brain and the spinal cord. So brain and the spinal cord in turn will result in the transmission of the information, that is motor information, from the central nervous system to the muscles of the leg concerned with the withdrawal of the leg from the source of pain or injury or prick. So that is how it functions. The details I will be discussing slowly so that you get oriented to the nervous system before you actually go on to the neural control of respiration. So the voluntary nervous system innervates the skeletal muscle. So subsequently, the leg is withdrawn, and that is the skeletal. So in addition to that, the sensory information, that is the receptors concerned with endocrines and other things will go to the central nervous system. That involuntarily results in the release of the secretion from the glands or the autonomic activity will be acting involuntarily. And then that causes the contraction of the cardiac muscle or contraction of the smooth muscle, etc. For example, you get excited and then the information goes to the brain from the receptors and nervous system. And that excitation will result in the release of the adrenaline or noradrenaline from the endocrines, the adrenal cortex, and that will be stimulating the heart so that the heart contracts vigorously. So this is how it functions. So in short, the sensory information goes to the brain, and then the motor information comes from this. So this is, it comes either to the voluntary muscles or involuntary muscles, that is exclusively the autonomic nervous system. So we are talking about the sensory nervous system and motor nervous system. And ultimately, it results in the basic unit of the central nervous system is a neuron. Just like the fundamental unit of the renal system is the nephron. So the fundamental unit of the central nervous system is the neuron. So all these activities will result in some influxes of ions. So it understands only one language, that is, you understand English, let us say. So the nervous system understands the language of fluxes of ions. That is, when the nerve cell is stimulated, it results in the fluxes of ions, and this fluxes of ions will result in the excitation of the neuron. And this neuron will result in the development of the action potential. So the action potential will be propagating from one side of the cell body to the axon and then terminates in the nerve terminal. So this nerve terminal may be a neuromuscular junction, or this nerve terminal may be a gland. So when it is a neuromuscular junction, it results in the excitation contraction coupling. When it is the gland, it results in the secretion from that particular cell. So that is when we are talking about the basic organization of the central nervous system. This is a voluntary one, and then that which innervates the gland is the involuntary one, that is autonomic nervous system. There are different types of the nerve cells, that is nerve cell, and then you have the Schwann cell. The Schwann cell is here, and then this winds around this axon, and then that results in the myelination. And this myelination is for the faster conduction of the nerve impulse from one end to the other end. So you have the other types of cells, namely the oligodendroglia. Oligodendroglia is that cell which is available in the central nervous system, which is mainly responsible for the myelination within the central nervous system. And then you have the astrocytes, you have the microglia, all those things are the different types of cells which you will be finding. So you're basically recollecting your events, that is, nerve getting stimulated, the basic unit of the central nervous system. Whether we are talking about the afferent nerve or the efferent nerve, it is a basic nerve that results in the excitation. And then if it is a voluntary one, it is resulting in excitation, contraction, coupling. And if it is a neuron, it is called excitation secretion coupling. And if it is a gland also, it is excitation secretion coupling. That is, the neurotransmitter is released at the nerve terminal. This neurotransmitter combines with the receptor in the post-synaptic cell. And that results in, again, initiation of the action potential in the post-junctional membrane. So always try to practice the diagrams, and then this practicing diagrams and then representing the same thing when you are writing the 3D paper is very important. So in continuation to what I am saying, this is the ion that is causing enters into the post-junctional membrane. That is, the fluxes of sodium ions enter through the sodium channel, and then that results in the excitation. Suppose had there been not the sodium ions, and then it's only the negatively charged chloride ion, that results in more negativity inside the cell membrane. Normally, inside the cell membrane is minus 70 millivolts. Whereas the further entry of the negatively charged chloride ions will result in further negativity, and that results in what is called the hyperpolarization. So when this occurs at the cell level, it causes inhibition. So normally in the post-junctional membrane, it rises from minus 70 to minus 50, and then you have the local potentials, and after the local potential, there will be spike potential, and then you have the development of the action potential. So this post-synaptic neuron can be either excitatory or inhibitory. So the post-synaptic neuron can be either an excitatory neuron or inhibitory neuron, depending upon the type of electrical changes which are occurring here. So we are just trying to revise what you have been discussed in the earlier class of the normal physiology. So you have sometimes they may ask you, what are the excitatory neurons and what are the inhibitory neurotransmitters? So these are the glutamate, norepinephrine, and dopamine are the excitatory neurotransmitters. And the inhibitory neurotransmitters are GABA, serotonin, and also dopamine. So we are just talking about the negativity inside the cell membrane. So all the cells have negativity inside the cell membrane, that is about minus 70 millivolt. And as a result of the entry of the positively charged sodium ions into the cell, so this negativity changes from minus 70 and gradually as a positively charged ion enters into the cell through the sodium pore, sodium channels. So this negativity will become less negative or more positive. So minus 69, minus 68, so on and so forth. And minus 55 millivolts is the threshold level. So from threshold level, as it reaches the threshold level, what happens is the cell membrane opens up more sodium channels. That is more in the sense the cell membrane transforms itself into a more populated sodium channels. And there will be a gush of sodium ions into the cell. And that results in the depolarization. And then this particular thing is called the spike potential. And it reaches the peak at about plus 30 millivolts, up to plus 30 millivolts. And then again, it comes back to the normal resting membrane because of the sodium potassium ATPase activity and channels. And then it is called repolarization. And then whenever you are talking about the hyperpolarization, there is the closure of the sodium channels, and so potassium gates remain open for longer than what is needed to restore the resting state. The neuron is insensitive to the stimulus and depolarization during this time. That is, sodium channels are decreased and then the potassium channels leak. And this is another picture of the nerve cell. So you have this nerve cell, and this is the nucleus. I just want to remind you at this point of time that is, cell is having a nucleus and the nucleolus. Every cell does have this. And then you have the axon hillock, and then you have the nodes, internodes, and then this channel bifurcates, and then this divides into a number of things, and this comes in contact with another nerve cell and then form a synapse. So this is the postsynaptic structure, and then these are the dendrites which are coming out from the cell, which make contact with another neuronal cell. So you have the central nervous system consists of the neuronal network. So all these mainly that results in the entry of nerve impulse from one nerve cell to another nerve cell. So this is about the brain picture, and then in the cross section, and you have these. They are all the brain structures, and this is the medulla. This is medulla here, and then pons and medulla. This area is of utmost importance at this point of consideration in the neural control of respiration. Other parts include the cerebellum, and then this is the brainstem. And then you have the thalamus and hypothalamus, which constitute what is called the diencephalon. Sometimes we just mention the diencephalon that infers that it is the hypothalamus and thalamus. They are the different parts of the brain. I think you are familiar with this terminology, what's it. So this is a better picture, and then this is the brain, and then this is the corpus callosum here, and then these are the lateral ventricles. These are the lateral ventricles, and this is the third ventricle, and then this is the fourth ventricle, and then this continues down the spinal canal. So, in short, you have the brain and you have the coverings to the brain. You have three coverings to the brain. That is Jura matter from without inwards. There are some terminology which means from outside to inside without inwards, we'll call. So you have the Jura matter, and then you have the arachnoid matter, and then you have the Paya matter. I'll discuss again in the later part. Jura, arachnoid, and Paya matter. This closely invests, covers the brain. And then you have, this is the pons, and the continuation is the medulla. And this is the pituitary gland. This is the pituitary in the sphenoid bone of the sella, that is sella turcica. And then you have the, here there is a venous sinus which is traversing throughout. And this is called the superior sagittal sinus. You open any textbook and then see the diagram and get familiar with that. And this is the choroid plexus. Normally what is happening is, the cerebrospinal fluid is formed in the choroid plexus. There are some blood vessels in the lateral ventricles. In the cerebral hemispheres, you have two lateral ventricles in which you find the blood vessels. These are the choroid plexus. Mostly the cerebrospinal fluid is synthesized in the choroid plexus. So from the choroid plexus, you have the two lateral ventricles, and then it goes into the third ventricle. Lateral ventricles, and then it is entering into the third ventricle, and then the CSF will flow into the fourth ventricle, and then into the central canal. So the ependymal cells, the other neuronal cells, also elaborate some amount of cerebrospinal fluid, and this will be released into this. The cerebrospinal fluid will be flowing here, and then it is passing down, and it moves in the it just comes out. You have comes out, and then as I mentioned, we have three layers to the brain. That is the Paya matter. Paya matter is a thin membrane which closely invests the brain from outside, and then you have the arachnoid, and then you have the Jura matter. Arachnoid matter is very close to the Jura matter. That is, Jura matter is on the inner side of the skull bone. So arachnoid matter is close to the Jura matter. So technically it is a potential space, but it is not the space between the Jura and arachnoid. And Jura, since it is very thick, they call it as pachymeninges. Pachymeninges, that is thick meninges. And then the arachnoid covering and then the Paya covering together is called lepto. So normally you have the blood-brain barrier. So the blood-brain barrier, all the elements of the blood do not enter into the cerebrospinal fluid. All the elements of the constituents will not enter into the filtered by ultrafiltration by the lateral plexus blood vessels. So that particular thing is called blood-brain barrier. You have the sodium ions, you have the chloride ions, you have the urea and other elements into the cerebrospinal fluid, and some amount of proteins also. So what happens is bigger sized things will not enter into the cerebrospinal fluid, and all the drugs will not enter into the cerebrospinal fluid, and even the hydrogen ions also do not enter into the cerebrospinal fluid. So in the chemical control of respiration, it is the carbon dioxide which is permeable through this blood-brain barrier and enters into the cerebrospinal fluid. There it dissociates, and then it forms hydrogen ion and the bicarbonate ion. The hydrogen ions will be stimulating the chemoreceptors in the respiratory system. So this is the mechanism that is operating. So normally the blood CSF will be formed, and then this will be going via the central canal, and then it comes out, and then it is present here, and it bathes all the parts of the brain, gives nourishment, gives buoyancy, gives cushioning, and prevents any shock to the central nervous system. So after circulating and after giving nourishment to the brain and the spinal cord, it is traversing in the subarachnoid space between the arachnoid matter and then the pia matter. And then it bathes. And then ultimately, there is what is called the arachnoid villi. Arachnoid villi are there. The arachnoid villi will be absorbing the cerebrospinal fluid, and then ultimately enters into the superior sagittal venous sinuses, and then goes into the circulation. So this is how the cerebrospinal fluid circulation is happening. Why I intend telling you at this point of time about the cerebrospinal fluid, brain, and the other thing is, so the changes in the hydrogen ion concentration consequent to the entry of the carbon dioxide into the cerebrospinal fluid blood-brain barrier, and then passes through, it will be stimulating the central chemoreceptors present, and then which in turn will be altering the respirations. And here I just want to tell you, please note down if you can, that is brainstem is the medulla and pons. Pons and brains, medulla are together are spoken of as the brainstem, from which you have the flowering part, which is the cerebrum, from which you find the flowering or the mushrooming part, namely the cerebrum, is the brain. And then somewhere much higher, you find inside, you find the thalamus and hypothalamus. And together, thalamus and hypothalamus are spoken of as diencephalon. Together, note down. Diencephalon means thalamus and hypothalamus. Cerebrum spreads over the diencephalon. And as I was telling you about the lateral ventricles, there are spaces within the brain substance of the right cerebral hemisphere and left cerebral hemisphere. Those are the lateral ventricles. Lateral ventricles have a rich blood supply, namely the choroid plexus, and there the cerebrospinal fluid is formed. And this cerebrospinal fluid, the lateral ventricles enter into the third ventricle, two lateral ventricles, and then it enters the third ventricle. From there, it enters the fourth ventricle. Here, there are for anatomically importance. So, lateral ventricles enter into the third ventricle. The lateral ventricles enter into the third ventricle through what is called foramen of Monro. Foramen, that is the opening, that is called foramen of Monro, M-O-N-R. So, whenever in the examination, in anatomy, they mention about foramen of Monro, you have to mention about that. From the foramen of Monro, is the third ventricle. It enters into through the cerebral aqueduct, enters through the cerebral aqueduct into the fourth ventricle, into the cerebral aqueduct, into the fourth ventricle. So from lateral ventricles, foramen Monro, and then this is the cerebral aqueduct. These two things you must remember. You should also remember that the thalamus and hypothalamus together are spoken of as diencephalon, over which you have the growth of the brain, that is the cerebral hemispheres. And you have the brain stem, that is the stem, something like the plant stem. You have the medulla and pons. The medulla and pons are of utmost importance for us as far as the control of respiration is concerned. So this is the another variant of the cerebrospinal fluid. You have the lateral ventricle here, and then so there are different anatomical parts. That is the lateral ventricle, and then the third ventricle, the cerebral aqueduct, and then you have the fourth ventricle, and then you have rich choroid plexus. Try to practice this diagram also. So the opening in the spinal cord is central canal, or canalis centralis. Sometimes it is. So you should also remember the spinal cord, as it is extending down. there is some mm, uh, enlargements in the thoracic area and in the lumbosacral area. This is mainly because of the presence of the upper limbs and the lower limbs. And it terminates, the spinal cord terminates at the level of the first lumbar vertebra. So the lower border of the first lumbar vertebra, where the spinal cord is in the form of a cone, inverted cone. And this particular portion is called conus medullaris. So this particular portion of the spinal cord is conus medullaris. You try to understand at this point of time, that is, the gray matter is outside in the brain and the gray matter is inside in the spinal cord. This point you must remember. The gray matter is outside in the brain and the gray matter is inside in the spinal cord. This point you must understand. So these are the different layers. You have the superior sagittal sinus here, and you have the subdural space, and this is the arachnoid space where you have the projections, and this one is the arachnoid villi, through which the cerebrospinal fluid is absorbed and then goes into the venous sinuses. So this venous sinuses will be draining and then getting into the venous circulation and enters into the right atrium. So these are the different parts of the skull. You have the skin. You have a visual picture. You have a visual picture of all these things so that you will be able to understand things better than memorizing. So you have in the order from without inward, skin, periosteum, the skull bone. Periosteal is meningeal layer. You have the dura mater. Dura mater is having two different things. Histologically, you can see that. The periosteal one and the meningeal one. And then you have the arachnoid matter, and then this pia mater. All the blood vessels which supply the brain are innervated and present within the pia mater. And then you have the arachnoid villus, which we mentioned. And these two cerebral hemispheres are separated by what is called false cerebri. False cerebri, a longitudinal fissure is there, through which the thing will be dipping down. So I was talking about the meninges, just for noting down. Outer one is the dura mater, the middle one is the arachnoid, and inner one is the pia. So dura mater is what is called, it is also called pachymeninges. Pachy means thick. For example, whenever you are talking about pachyderma and other things, that is prefix means pachy means thickness. And these are arachnoid matter and the pia mater, they are together are called the leptomeninges. So for the clinical importance, whenever there is inflammation to the coverings of the brain, so this is called meningitis. Normally the bacterial thing and then the viral things will be causing the inflammation of the meninges, and that results in series of complications in the children and adults. So that particular thing is called meningitis. Meningitis. Itis means normally infection. Whenever you have the suffix term itis, that is called the infection, the meningitis. So normally tuberculous bacilli will be entering into the brain and involve these things. So mostly you find all these tuberculous meningitis, viral meningitis, that is type of, most of the things you must be seeing in the news items, that is children dying, mainly because of the viral meningitis. So these are transmitted from pigs, mosquitoes, etc., and then ultimately resulting in the meningeal infection, and that is called the meningitis. So if it is caused by the bacteria, it is called bacterial meningitis, and if it is because of the viral thing, it is called viral meningitis. Normally, these anatomical picture of these things is important because you have to tap fluid from different parts of the spinal cord. That is, cerebrospinal fluid has to be extracted to know about the actual damage. So this is the picture, the brain, spinal cord, and then this is the conus medullaris here. And then ultimately it is, there will be number of rootlets coming out from the conus medullaris, and that resembles the tail of a horse. Hence it is called cauda equina. These are the things which are commonly asked in the viva in anatomy or physiology. That is the terms which means you know about the conus medullaris, you know about the spinal cord, you know about the rootlets which are coming out from the terminal end of the spinal cord. That infers that. So here you must remember the spinal cord terminates at the level of L1, and then there you find only the dura mater, arachnoid mater, the clear. And this is devoid of the nervous tissue or the spinal cord. So the brain, spinal cord, the dural membrane, and then here they are depicting about the sacrum. So here in the brain, you find different types of cells. So if they ask what are the different types of cells in the central nervous system, you have to mention all this. This is the microglia, and you have the astrocytes. Astrocytes are so named because of the shape of the cell. And then this is the blood capillary. They are closely investing the blood capillaries, and they get nourishment from the blood vessels, and these astrocytes will in turn will be giving nourishment to the neuronal cells. And then you have the ependymal cells. The ependymal cells are mostly secretory in nature. Also in the spinal cord, in addition to the presence lining the canal, it will also secrete the cerebrospinal fluid. The oligodendroglia are mainly responsible for the myelination in the central nervous system, whereas the myelination in the peripheral nervous system is by the Schwann cells. So you have the lateral ventricle, and then the third ventricle and the fourth ventricle, and then passes down, and then it circulates and ultimately passes through the different areas of the coverings, and then goes to the arachnoid villi, and then into the superior sagittal sinus. So here it is depicting that is CSF is secreted by the choroid plexus, flows through the interventricular foramina into the third ventricle. Choroid plexus in third ventricle adds more CSF, and then the CSF flows down the aqueduct to the fourth ventricle. The choroid plexus in the fourth ventricle adds more CSF. CSF flows out two lateral apertures and then one median aperture. CSF fills subarachnoid space and bathes the external surface of the brain and the spinal cord, arachnoid villi, and are reabsorbed into the venous sinuses. So if you can know about this pathway, you can write a lot of questions. So this is about the circulation. Again, superior sagittal sinus. So you try to read about the osteology and then about the skull and different parts. This goes a long way in understanding about the anatomy of the skull bones and the brain. So from the spinal cord, as I said, the gray matter is inside, and then the white matter is outside. White matter is nothing but the axons and dendrites, which will be normally seen. So this is only the myelinated nerve fibers, nerve paths. Normally, you have the anterolateral aspect, you have the nerve rootlets. They together form a single nerve, and then they form a common, you have the nerve rootlets here, and then the anterior nerve rootlets, and then the posterior nerve rootlets. They form a bundle, and then these bundle together unite and form a spinal nerve. And the posterior root, you have the posterior root ganglion. This word you must remember. So in short, what is happening is in the spinal cord, from the anterolateral aspect in the anterior side and in the posterior side, you find number of rootlets in each spinal segment. You have the anterior nerve rootlets joined together to form a nerve and a root, and then both the roots will combine together, and that the posterior end, that is the slight bulging, that particular thing is called posterior root ganglion, and ultimately become the spinal nerve, which exists from the vertebral canal, vertebral lateral one. So, and this is covered again, the entire part of the spinal cord is covered by the pia matter, the arachnoid matter, and then the jura. You have the jura matter here, and then you have the pia. The pia matter does have the blood vessels, everything embedded within that. So the brain has gray matter outside, and the spinal cord has gray matter inside in the form of H. So I was introducing you to new terminologies, namely the afferent and efferent. The normal textbook, they'll be just mentioning about the afferent and efferent. Afferent or the sensory division transmit impulses from the peripheral organs to the central nervous system. And efferent or the motor division transmit impulses from the central nervous system out to the peripheral organs to cause an effect or action. Afferent neurons are the sensory nerves carrying the nerve impulses from the sensory stimuli towards the central nervous system and the brain. So there is a there is some technical thing here. Efferent neurons are the motor nerves carrying nerve impulses from the central nervous system and the brain. This is in response to the sensory stimuli. Note down this is very important. Generally, we are going to talk about the afferent and efferent. Afferent terminates in the central nervous system, and efferent is coming out from the central nervous system and innervating the voluntary and the autonomic nervous system, namely the glands or the heart or the other parts. So normally they'll be asking about the afferent and efferent thing. So I was just telling you about the entry of the information into the spinal cord. That is, sensory information will be passing through, and then you have the motor information coming out. So the motor information to the autonomic nervous system and somatic thing, that is the motor nucleus, and it will be terminating. This is how the pathway goes. So the same thing, the afferent information coming, and then the efferent information going to the motor. So normally, as I said, the spinal cord ends at the level of L1, lower border of L1, and subsequently you find only the covering, namely the dura mater, arachnoid matter, and then the pia mater, which closely invests the spinal canal in the vertebral column. So normally, the cerebrospinal fluid is taken out from the from L2 and L3. So the patient is asked to be on a lateral position and then in the curling position, and then you give local anesthesia to the back of the individual in the lower portion, in the lumbosacral area, give local anesthesia, and then you pass a needle, 22 bore needle between the intervertebral disc and then go into the spinal space. And then you will be collecting the cerebrospinal fluid for analysis in suspected cases of meningitis and other conditions. So these are the different parts of the cerebrum. They are the different parts of the cerebrum. You have the central sulcus. So whenever you're talking about the invagination, it is called the sulcus. Whenever there is invagination into the brain substance, it is called the sulcus. And the elevated portion you call as gyrus. So you have to understand the word sulcus and gyrus. Gyrus is the elevated portion. Sulcus is what it invaginates down. So different areas are given different names. That is, the precentral is sensory and post-central is motor. And then you have the parietal lobe, and then you have the occipital lobe. And this is the frontal lobe. And then you have here the pons and medulla. In the normal supine position, normal vertical position, you find the brain in this orientation. And behind that, you have the cerebellum. And then you have the pons and medulla. That is part of the diencephalon. So there is a representation of the body in the precentral gyrus and the postcentral, both in the sensory and then the motor area. That representation of the body is called sensory homunculus, and motor homunculus. So in the cerebral cortex of the brain, you have the representation of the body based on the sensory input to the brain. And this particular thing is called sensory homunculus and motor homunculus. All the information from the sensory organs will be going to the portion. And there is the representation, something like a map of India. There is a representation of the body also in the brain. That is called sensory homunculus and motor homunculus. When that particular area is stimulated, that results in the initiation through the nerve fibers originating from the cerebral cortex to the terminal organs or the muscles. So in the central nervous system, there will be some terminology which you must know. That is nucleus. So nucleus of a cell is different from nucleus, the term nucleus, which you mention in the central nervous system. In neuroanatomy, a nucleus, that is plural form is the nuclei, is the cluster of neuronal cells. That is, if a number of nerve cells are there together, you call it as nucleus. This nucleus is quite different from what you talked about the nerve cell, individual nerve cell. This is collectively in the central nervous system, a cluster of cells is called the neuron. Neurons together in the central nervous system is called the nucleus. So this nucleus in the central nervous system are located deep within the cerebral hemispheres and the brain stem. The cerebral hemispheres, you have the cerebral cortex, and the brain stem, you have the medulla and pons. So this nucleus in the medulla and pons, which have a respiratory control, V is the nuclei. So that's the reason why I'm trying to tell you about this. Nuclei are connected to other nuclei by in the form of tracts, the bundles of axons, nerve fibers extending from the cell bodies. And there is another thing, the ganglion. So I just go back to the thing. So this is the posterior root ganglion. You are talking about the posterior root ganglion. That is, the sensory information is entering into the spinal cord, and then the second order neurons coming, and then it is going to the motor neuron. At this end, there is a swelling. This particular thing is called ganglion, for which I am trying to derive at this particular terminology. Ganglion. So generally in the Viva, they will be asking about these things, and then you should not get confused here. The ganglion sometimes also refers to a small cystic tumor connected either with the joint membrane or tendon sheath. So sometimes when you go to the orthopedics subject and then you talk about the ganglion, that ganglion is different. And the ganglion which I have shown just now is different. A small cystic tumor connected either with the joint membrane or the tendon sheath. Joint membrane or a tendon sheath is the cystic tumor. That is the ganglion. I'll show you the picture later. The mass of the nerve tissue containing cell bodies of the neurons external to the brain or the spinal cord. So this is the mass of the neuronal cells outside neuronal cell bodies. So the mass of the nervous tissue containing the cell bodies of the neuron external to the brain and the spinal cord. These are outside the brain and the spinal cord. These are outside the brain and the spinal cord. This point you must remember. The ganglion is outside, not within. So this is the ganglion, which normally you see. If you can see the people, observe the people, they'll be having the ganglion on the wrist, most of the people. And sometimes this dissolves on its own. And sometimes if it is obstructing to the movements of the tendon, it is surgically removed. So this is the ganglion in the bones, very close to that. This is the ganglion. So when you are referring to the ganglion with reference to the clinical thing, it is the mass. And you have the outside the brain and the spinal cord, you find the elevations, which is the ganglia. So this amount of introduction is necessary for thorough understanding of the neural control of respiration. So you have the higher centers in the brain, namely the from the cerebral cortex. So you have the cerebral cortex, the limbic system concerned with the emotions, and the hypothalamus also is concerned with the emotions of the individual. For example, the individual gets excited, that is as a result of some positive news or a negative news. It is the hypothalamus and the limbic system are stimulated, which in turn will be stimulating the respiratory centers which are present in the diencephalon, that is the medulla and pons. And further, the cerebrospinal fluid, the changes in the chemistry of the cerebrospinal fluid, and then the cerebrospinal fluid as it is getting circulated into the cerebrospinal fluid pathway, it will be resulting in the central chemoreceptors, and this in turn will be resulting in the stimulation of the respirations. So the chemoreceptors and baroreceptors and aortic sinuses, you are familiar with that. We'll discuss again in the next class when I'm dealing with the chemical control of respiration. So when the lungs are stretched, the receptors will carry information to the respiratory centers. So normally under quite resting conditions, the inspiration and expiration goes normally. widely. This is an important diagram. Try to note down. Nerves related to nucleus, tractus solitarius. So we are talking about the nucleus. The term nucleus means the collection of the nerve cells. And this thing is called nucleus tractus solitarius. That which is mentioned in the violet color here is the nucleus tractus solitarius. And this is the pons, and lower down is the medulla. So just also note down, it's a mnemonic wherein nucleus tractus solitarius. Nucleus because it is the collection of the nerve cells. Tractus because it is in the form of a tract. And solitarius because it is isolated. So the term. So normally in anatomy, if you can remember the terminology, it gives a lot of information. You concentrate on that. So nucleus is the collection of the nerve cells, and then the tractus because it is in the form of a tract, and then solitarius because it is isolated, unconnected to other parts. And you should also remember here, N means ninth. Ninth is the glossopharyngeal nerve. Nine is the glossopharyngeal nerve. You have 12 cranial nerves, out of which ninth one is the glossopharyngeal nerve. And the 10th nerve is the vagus nerve. 10th nerve is the vagus nerve. And nucleus tractus solitarius is the 7th nerve. 7th nerve also innervates, and that is related to the nucleus tractus solitarius. Why I am telling you at this point of time is all the chemoreceptors, the sensory signals are carried by the glossopharyngeal and vagus nerve, and they terminate in the nucleus tractus solitarius, near and in the nucleus tractus solitarius. In addition to that, the dorsal respiratory group is also present within the substance of the nucleus tractus solitarius. The dorsal respiratory group is also present within the substance of the nucleus tractus solitarius. Further, it is interesting for you to know that you have another set of topics which you'll be dealing with in the special senses. The special senses in the sense, taste, smell, gustatory sensation, and then the visual, hearing. All these things come under special senses, which we'll be dealing with in a separate group of classes after the central nervous system. So in which the tongue is supplied, the sensory information to the tongue, anterior 2/3 of the tongue is separate, and then the posterior 1/3 of the tongue is separate. Because embryologically it develops from the occipital somites. Embryologically it develops from the occipital somites and then migrate up, and then it becomes the tongue. So anterior 2/3 of the tongue is having a different embryological origin, and then the posterior 1/3 of the tongue is having different embryological origin. So along with that, it carries the nerve, that is the glossopharyngeal nerve, and then the vagus nerve supply. And the 7th cranial nerve carries the sensory stimulation from the anterior 2/3 and posterior 1/3, that is entire tongue area. So this sensory pathway, the gustatory pathway from the tongue, will go via the anterior 2/3 of the tongue, glossopharyngeal, vagus, and then it moves up with the medial lemniscus and then terminate in the post-central gyrus, which is the sensory cortex. So this is the gustatory pathway. So all these chemical sensations will be going and terminating the nucleus tractus solitarius. Hence, I was referring to all these things in an elaborate manner. So you should also remember that in the chemical control of respiration, the peripheral chemoreceptors, which are present in the carotid and aortic area, will terminate in the nucleus tractus solitarius. That is, it is a chemosensitive area in the brain. So this light blue colored one is tractus solitarius. So you have the respiratory centers and then different nucleus of the cranial nerves here, trigeminal nerves. So whenever you get bored, you try to open your textbooks and scribble your this diagram and note down where exactly you find nucleus, tractus solitarius. At least do one thing at a time so that you will remember forever. And this is the nucleus, tractus solitarius sensory nucleus is located in the dorsolateral medulla oblongata and lower pons, generating and synchronizing the peristaltic activity of the upper gastrointestinal tract during the swallowing. So it also generates and synchronizes the peristaltic activity in the upper part of the gastrointestinal tract during swallowing. Inhibitory, that is the GABA and excitatory glutamic transmitters, is responsible for the sequential motor pattern of the peristalsis. Peristalsis is an important term which you should concentrate when the gastrointestinal tract is being discussed. The number of candidates are 115. Sorry, attendance is absent marks. So information processing is done at the nucleus tractus solitarius and the respiratory rhythm generation. Nucleus tractus solitarius relays information from the primary visceral receptors to the central nervous system, involved in the reflex control of the autonomic functions. Nucleus tractus solitarius is the first synaptic station of the cardiorespiratory afferent input, including the peripheral chemoreceptors, baroreceptors, and pulmonary stretch receptors. This is an important point. This you can just note down. The nucleus tractus solitarius is the first synaptic station of the cardiorespiratory afferent inputs, including the peripheral chemoreceptors and baroreceptors and pulmonary stretch receptors. Coming to the respiratory centers, madam. This is 115. Yes. People who are absent, who left, and then give me the numbers. So you have centers, note down, this is an important diagram for the respiratory centers. That is a pneumotaxic center. I just mentioned about these centers in the beginning. Pneumotaxic center, nucleus parabrachialis, medialis, and nucleus fused. Not necessary, just remember pneumotaxic center at your level. And below that, you have what is called the apnostic center. You try to know about this spelling also. And these two are present in the pons. These two are present in the pons. The pneumotaxic center and the apnostic center are normally present in the pons. And the dorsal respiratory group here, dorsal respiratory group, that is nucleus tractus solitarius. And this is the ventral respiratory group. Botzinger complex. Just remember the term I'll discuss with you. Botzinger complex. Nucleus retroambiguous, caudal and rostral portions. And these are present in what is called the medulla oblongata. These are present in what is called the medulla oblongata. So in the cross section, you find pneumotaxic center here, and then you have the apnostic center here. And you have the dorsal respiratory group, wherein the sensory input coming from the vagus and the glossopharyngeal nerve. And then from this, because of this sensory information and then relay and then development of all the activities, this results in the output, which goes to the abdominal muscles and then the internal intercostal muscles for the inspiratory act. And that in turn, because of the stretch and all that, will be resulting in the stimulation of the ventral respiratory group. And that results in what is called the expiration. And you just identify pre-Bott-Zinger complex, which is a red colored one here. I'll just talk about this later. Above which you find the apnostic center. Apnostic center will receive continuously the inhibitory impulses from the pneumotaxic center. In short, pneumotaxic center is inhibitory to the apnostic center. Apnostic center will be resulting in the stimulation of the dorsal respiratory group, that is the inspiratory center. And this in turn results in the efferents coming out from the vagus, afferents coming out from the vagus getting stimulated, and then efferents going from the abdominal and intercostal muscle. Vagus, dorsal respiratory group, coming out to the abdominal nerve innervation. So these are the muscles of inspiration. Normally, it results in the stimulation of the external intercostal muscles. So subsequently, the flow from the apnostic center and dorsal respiratory group will result in the stimulation of the external intercostal muscles, which in turn elevate the ribs, and then the diaphragm moves down, and the scalene, and then the sternum, the body of the sternum moves forwards. So there is a three-dimensional picture. Anterior posterior diameter is increased, and above downwards diameter is increased, and then because of the movement of the diaphragm, and then the overall space within is increased because of the eversion and elevation of the ribs by the action of the external intercostal muscles. Further, if in deep exercise or severe exercise, if the inspiratory act requires further action of the accessory muscles, it results in the action of the scalene muscles, which elevate the first and second ribs also. And in general, the expiration is normally the elastic recoil of the thoracic cage, but in situations where expiration got to be active, it results in the stimulation of the ventral respiratory group. So the ventral respiratory group are stimulated, and this stimulation will result in the action of the expiratory muscle, internal intercostal muscles, and then it results in expiration. It results in removal of air from the lungs. So actually, there is an oscillatory model between the dorsal respiratory group and the ventral respiratory group, or in other words, the inspiratory group and then the expiratory group. So this you can analog this and then compare to that of a pendulum. So in a clock, the pendulum moves from one end and then moves to the other end. This oscillatory model is there between the inspiratory center and the expiratory center, or the dorsal respiratory group of neurons and the inspiratory group of neurons. And then this internal intercostal muscles will recoil, and then the sternum moves back and the diaphragm moves up. So diaphragm, internal intercostal muscles depress the ribs, and then diaphragm moves up and then relaxes. And then you also have the transversus thoracic muscle, which depresses the ribs, and the inferior part of the sternum, the sternal body, moves backwards. So these are the mechanics which are occurring in the neural control of respiration. So you have the breathing control centers, and this in turn will be resulting in the nerve signals to the muscles of inspiration and the muscles of expiration. Note down, this is very important. So pre-Bott-Zinger complex. pre-Board-Singer complex. So this pinkish one, which I mentioned in the earlier slide, is the pre-Board-Singer complex. What does it do? Pre-Board-Singer complex is the autonomic nervous system control of the respiration. It is initiated by the rhythmic discharge from the pacemaker cells in the pre-Board-Singer complex located in the medulla oblongata near nucleus ambiguous. So this exposure to the very rare terminologies will not interest you at this point of time, but it will go a long way in understanding the later part of your curriculum. So this something like, what does a pacemaker cell means? They say this is a cell which has the maximal speed. So this is called a pacemaker cell. So you have the pacemaker cells in the heart, that is the SCA node. And then similarly, you also have the pacemaker cells in the brain. So that is the pre-Board-Singer complex, that is which initiate the process of action of the dorsal respiratory group. So this pre-Board-Singer complex is mainly responsible for initiating the pace in the dorsal respiratory group. And this is very close to the nucleus ambiguous. These things you can learn in the later portion. Voluntary control of respiration is located in the cerebral cortex. Each and every word here that is mentioned is very important. The voluntary control of the respirations is located in the cerebral cortex. I have shown you the diagram of the cerebral cortex. I have shown you the diagram of the body representation in the cerebral cortex. So the particular area will be stimulated, and that stimulation will result in the contraction of the external intercostal muscles and internal intercostal muscles during the act of inspiration. And coming to the applied aspects, some of the things which you should know, I will discuss in the later part. Apnostic center excite the inspiratory center. Stimulation of the apnostic center leads to gradual increase in the firing rate of the So stimulation of the apnostic center leads to the gradual increase in the firing rate of the inspiratory muscles. And pneumotaxic center provides inhibitory impulses on the inspiration, prevents over distension of the lungs, maintain the alternately recurrent inspiration and expiration. Apnostic center in the pons increases the depth and duration of inspiration. Pneumotaxic center decreases the depth and duration of respiration. Cerebellum, hypothalamus, motor cerebral cortex, and limbic system also play a role in mediating respiration. These points, note down. The cerebellum, hypothalamus, motor cerebral cortex, and the limbic system also play a role in mediating inspiration. So, in short, about the genesis of inspiration, rhythmic discharge, apnostic center stimulation, inspiratory discharge, and C3, C4, C5, and T1 and T2 of the spinal cord. And all these stimulation will result in contraction of the muscles of inspiration. And genesis of respiration, inspiratory neurons in the medulla are stimulated. They stimulate the pneumotaxic center, that is upper pons, inhibit apnostic center, that is pulmonary stretch receptors in the lungs via the vagus nerve, stimulate the expiratory center, and apnostic center gets inhibited and stops to activate the inspiratory center. Again, the same thing. Dorsal respiratory group and the ventral respiratory group stimulation. You should know that inspiration is followed by expiration, where the normal ventilation is retained. So some of the applied aspects which you should know. Apnea is the transient cessation of breathing. Sometimes we should be familiar with these things. During apnea, there is no movement of the muscles of the inhalation, and the volume of the lungs initially remains unchanged. Eupnea is normal breathing. Tachypnea is rapid breathing. Normally, the adult breathes from 12 to 15 times per minute. In children, the rate is much higher. So you should know about apnea, eupnea, and tachypnea. Hyperapnea is breathing more deeply and sometimes faster than usual. It is normally during exercise or exertion. Tachypnea is rapid, shallow breathing when you take more and more amount of air per minute. Hypercapnia is building up of the carbon dioxide in the bloodstream, seen in chronic obstructive pulmonary disease. Remember this. Efferents from higher centers, the cerebral cortex, these are the different parts, cerebral cortex and cingulate gyrus inhibits respiration. The stimulation of these parts of the brain will result in inhibition of respiration. Corticobulbar and corticospinal tracts modify the activity of the respiratory muscles. How does hypothalamus and limbic system affect? It is pain and emotional disturbances. For example, you are in pain, you are weeping, your respiratory rate goes up. In temperature, because of the stimulation of the anterior portion of the hypothalamus, where you find thermoregulatory center, it results in increase in the respiration, that is tachypnea. So afferents from the proprioceptor, that is the proprioceptors or the receptors which tell you about the movement of the joint, also result in stimulation of the respiratory center, and then there is increase in the respiratory center. For example, you are running, that results in the stimulation of the proprioceptors in the joints, and that in turn will be stimulating your respiratory center, and then the respiratory rate goes up. Afferents from the pharynx, trachea, and the bronchi also will result in affecting your respiration. What exactly is breaking point? That is not the point where you break something. Breaking point is the point at which the breathing can no longer be voluntarily inhibited, and is called the breaking point. Breaking point is generally reached when the alveolar oxygen tension is 56 millimeters of mercury, or carbon dioxide tension is 49 millimeters of mercury. This is another commonly asked short note question, the breaking point. So this is the corticospinal pathway. I think it is too much at this point of time. And this is corticobulbar pathway. I think you can avoid this.